Thank you. The last thing that that Andre said to me was that we were talking about this conference, because we were talking about when we were going to see each other next, and he said, "I hope you're not planning on talking about necessarily things that I've worked on. You should talk about things that you're doing now, and if that's stuff that I've worked on, then that's great. But just talk about what you're most interested in right now." And that's what he wanted to hear, and so that's what I'm going to do. Now, that being said, Andre's work has had a huge impact on my trajectory. Two of my best papers involve directly stuff that he's worked on, hypergeometric systems and connections to commutative algebra. That was in some joint work of mine with Uli Walter and Laura Matusevich. And what essentially amounts to a 97-page combinatorial treatise on a beautiful 2-page paper by Andre on how to embed the representations of a type A quiver into a Schubert variety. That was joint work with Alan Knudsen and Mark Shimozono, and actually leads directly into in some sense, leads directly into the kind of things that I'm going to talk about today. But anyway, I think Andre would be amused by the direction that my experience with these topics has taken me in, and it's an honor to present these things to this audience. So this work is joint with a number of people. There's some stars and some daggers. So I'm really going to be talking about sort of two joint works. One is joint with the first is joint with Paul Bendich and Steve Marin and Sean Square. Paul Bendich is a sort of a between math and CS. He was a postdoc of Herbert Edelsbrunner's. Steve Marin is a statistician at UNC Chapel Hill, and Sean Square is in operations research at Chapel Hill. He's a student of Steve Marin and Scott Proven, whose name will again come up later in this talk. David Houle, the other papers, that's the stuff with the brains. And then the Flywing work is with Paul Bendich again, and with David Houle, who is an evolutionary biologist at Florida State. So this has inputs from sort of all over. So I'll start by defining persistent homology, and then I'll talk about barcodes. Fortunately for this audience, these topics are very they should be familiar, or they will be familiar after I cover them. You'll see why. And then I'll talk about brain trees. And then I'll show you some videos and things. Flywings, that's going to be analyzing images of flywings, in particular the veins in flywings, and the relation of that to evolutionary biology. And the methods that we'll use for that will be stratified persistence, which I'll talk about, including this notion of metric perversity, just like the perversity that you see in intersection homology, but done for sort of metric stratified spaces. And then I'll talk about some implications that this has for what biology should be doing. And then I'll talk about sort of stratified statistics in general, which happens when you're trying to sample from stratified spaces. And then I'll talk about some next steps in math and give a little bit of review of what we've done. So persistent homology. So Andre likes to define things first, and thankfully with this audience, in almost no other talk can I define persistent homology in 3 lines. But with this audience, I can do it. And if you give me a sequence of spaces, so just continuous maps between topological spaces, then I can make the corresponding quiver of the homology of these spaces. And that's what's called the persistent homology. It's sort of nothing more and nothing less than that. Now I'm going to assume that the homology is over a field so that I get, easy direct sum decompositions and these kinds of things. And I'm going to assume that all the spaces are tame, because for one thing they come up in computational purposes, and so the spaces are actually tame. And what I mean by that is that they have finitely generated homology. None of these complicated counterexamples to various things in topology. We're dealing with real, honest examples. So what kinds of how do you get persistent homology? What kinds of sequences of continuous maps do we get? Well, if you have any tamely filtered space, all I mean by that is that the filtered pieces are tame spaces. So I'm not having sort of lots of homology appear and then disappear. There can be lots as long as there isn't infinitely much appearing and disappearing. And all these 3 are examples of what happens when you have, are examples of this tamely filtered space business. So if you have, for instance, a function on a topological space, and I can look at the part of the topological space that is in the pre-image from, if you look at the sort of between negative infinity and some fixed time. So you should think that you're taking your space, and just like you have a moment map, you sort of squish the thing onto a line, and you look at all the sub-level sets. So you pick a level, and you look at everything that's beneath that. And then you let the level go up. You're going to be building your space. You do this in Morse theory, and often this is actually assumed to be a Morse function, but it doesn't really have to be for abstract persistent homology. It can just be a function, as long as there are only finitely many values across which the topology changes in some way, then you get persistent homology in this sense. And in the case where you're dealing with a function where the topology changes at certain times t, certain real numbers t, instead of indexing the quiver just by which t is it, as in like, so I can order them t1 through tm and say that, well, it's t1 or t2. Instead of saying it's the first or the second t, I can say it's actually the t itself, so that the edges of the quiver have lengths. And so it makes sense to say that if you have a homology class that sort of appears in a certain sub-level set, then it persists for a certain amount of time, and then it dies, for instance, if it's a component that was then joined to something else or something like that. So this is persistence in the sense of homology is persisting for a certain amount of time. That's what persistent homology is meant to evoke as a term. So that's a very common. This is, as I say, this is probably the most common way that people in persistent homology introduce it when they're giving a talk. But of course, I can just use quivers because a lot of people here have seen quivers. If you have a simplicial complex, then you can build it simplex by simplex in some order. That actually makes it a filtered complex as long as you add the simplices in some appropriate kind of order. And this allows you to say sort of precise things like when a homology class is born or dies, because you might have, for instance, the last simplex that makes a sphere, and then you'll have a two-dimensional homology class or something. In fact, any sequence of spaces can be reduced to the filtered space construction with a function on it by just using mapping cylinders. So these are sort of all equivalent ways of thinking of persistent homology. So I'm not going to get into the complicated history of this, but it was invented independently by a number of different groups with a number of different purposes. But in particular, this third group, the Adelsbrunner group at Duke, their paper from 2002 was part of one of the simultaneous. They were working on it earlier than 2002, but this particular paper also introduces efficient computational methods for doing these things. They were interested in a lot of three-dimensional geometry, computational geometry type problems like protein docking and these kinds of things. Now, a lot of other people have worked on persistent homology over the years. I want to particularly mention Carlson, because Gunnar Carlson at Stanford, he's been sort of key in a lot of the developments that connect persistent homology to potential applications, and also thinking of sort of the nice abstract mathematical ways of thinking about it. So I should say, by the way, that when I gave the outline at the beginning, each of those items on the outline was one slide, except for one. So when I say one slide, I mean one page like this. There's only one that involves a lot of slides. I think this talk has 82 slides in it, but most of those slides come in the brain trees part because they're just sequences of images of things growing and so on and so forth. So if you want to know where we are in the talk, that might help. So the barcode of a sequence of spaces or of a filtered space, x dot, the barcode is just the way it decomposes. So remember, persistent homology is a type A equioriented quiver. And so it decomposes as a direct sum of indecomposable summands. And the list of indecomposable summands is the barcode. And you can think of this however you like to think of it. You can call it a barcode. You can call it a lace array, which was how Alan and Mark and I called it in our 97-page paper on quiver cycles. We called them lace arrays because we were doing lacing diagrams and so on. But it's just the multi-set of line segments, and it's one for each indecomposable summand. So here's a picture of a barcode, and this is an actual data point in the study that I'm going to show you about the brains. So this is all, in fact, zero-dimensional homology. But I've put, but the computer put the line segment that goes from t to t prime. It started it at the point, so x coordinate t, and so it goes from the diagonal up from t to t prime. That is, you can think of this as a homology class is born at this time, and then it persists until this time, and then it dies. In this particular case, it's a component is born, and then at some point it's joined back to the rest of the stuff, and then that's why it dies. And the point is that a component can't die before it's born. That's why everything is above the diagonal in this picture. It's usually, as I say, customary to separate the barcode into pages, one for each homological degree. In this particular case, we're talking about persistent homology of a tree, or actually, I think it might be a forest. But in any case, it's only got zero-dimensional homology at any stage. So in fact, it's not necessary to label it with a homological degree, but it is usual to separate the pages into various homological degrees. So this is computable for simplicial complexes if you order the simplices compatibly. So a compatible ordering would be if you have a filtered space. So typically what you'll do is you'll have a space, you'll triangulate it, and then you'll order the simplices in such a way that all the simplices in each filtered piece come before all the simplices that are after that filtered piece. And furthermore, when you add a simplex, you don't add a simplex until you've added all of its faces. And then easy, row reduction type algorithms can compute persistent homology efficiently, actually quite efficiently, in polynomial time. But even, yeah, it gets bad when the dimensions go up. But for instance, this was computed automatically from a brain example. So are there any questions about persistent homology as a, Sergey?
Do you understand it correctly that here the multiset is actually a set?
Yeah, so the question is, do you understand correctly that the multiset is a set? And the answer is yes, because this comes from real honest data. So from real honest data, the points, the spaces, they're all sort of generically placed. I mean, the chances that 2 points will that some two classes will be born at exactly the same time in real data is basically zero. In sort of mathematical examples, of course, the multiset really is a multiset, and you have lots of them being born at the same time and dying at the same time. But when you're actually doing data analysis using these kinds of things, you do really expect to get a diagram where they're all got distinct birth times and death times. Well, sorry. Sometimes there's a lot of things born at zero, for instance. In fact, this bar might be a multiset here, but most of the time you expect them to be distinct birth and death times. Yeah.
This is probably a stupid question since I don't know quiver homology. But in the definition of persistent homology that goes elementarily, I remember there being some lag. Like you take the cycles at time t, modulo the boundaries at time t minus something. How does this exactly translate into this quiver thing?
Yeah, so the question is, how does the usual definition of persistent homology in terms of cycles at time t modulo boundaries at some previous time, how does that translate into the quivers? If you look at the homology at some time, then that's going to be cycles mod boundaries. But everything that was, you have to think about what is a boundary at time t and what it means to be a cycle coming from a filtered piece that's lower down. It's just a matter of, so when you look at the induced maps on homology, to say that a cycle is born at time t means that it's a cycle in the tth filtered piece, but it didn't come from a cycle that was in one of the previous filtered pieces, and it also is not a boundary in the tth piece, means that it didn't come from the boundary of something earlier. There's a sense in which you should sort of forget about the quiver part and look at just the barcode part. So in the end, usual persistent homology is going to give you this barcode. It's going to say some classes are born. There's a class that's born at a certain time. It persists for a while, and it dies at a certain time. Viewing this thing, quiver homology, viewing this thing, this persistence diagram as coming from a quiver is just another way to arrive at that barcode. And it's giving you equivalent information. The decomposition as a direct sum is canonical. The number of sum ends is canonical. And so the data that you're getting out of it is the same either way. But I don't want to go into too much of the detail of how you compute these things. But more or less, this can be used to compute the persistent homology in the quiver sense and in the persistent homology sense. Are there any other questions about this before I go on? I'm going to show you some examples next. That's what's going to happen. Okay, so persistence on embedded trees. So these, the goal of this is to do statistical analysis on images, taking three-dimensional geometry into account. The three-dimensional geometry is, so the images are going to be brain images. So this is a magnetic resonance angiograph of Steve Marin's brain. I can tell you whose brain it is because he's a co-author, and he was he took part in this study by Elizabeth Bullitt, who's actually another co-author of mine on a different paper. But there's a big group doing these kinds of things. Anyway, so this is an MRI image, but it's an MRI image whose purpose is to look at flowing things. So the bright white stuff that you see. So, for instance, over here and over here, these are blood vessels. These are arteries, actually, where the blood is flowing robustly. And what's going to happen is I'm going to give you a sequence of these images, and you'll see that the arteries sort of move. So this is the brain scan is moving up through the brain, and you can see the arteries how they sort of track through, right? And you could think if you were really smart, you might be able to figure out how to draw a three-dimensional picture of the blood vessels, given that these are the two-dimensional slices, and in fact, that's what they do. So this is a three-dimensional picture of the blood vessels in Steve Marin's brain. And just to give you an idea of what this looks like, I'm going to give you just five or six rotated images. Is it clear what's going on? That is an that's a fair and excellent question. The question is, what's the topology of this thing? So this is not actually so these are only arteries. If we included arteries and veins, then it would have lots of circles and cycles and so on. But since this is only detecting arteries, it's sort of detecting them until the resolution goes too low, and then it's not seeing capillaries. It's really only seeing sort of not macroscopic exactly, but these are down to the maybe slightly sub-millimeter resolution, and then they disappear off of the MRA. So you're seeing something that is definitely it definitely has no loops, but it's not necessarily connected, and in fact the number of connected components can vary from person to person. The most common is that people have four. There's a circular blood vessel in your neck or at the base of your brain called the circle of Willis, and there are major arteries coming up out of it, and the majority. I won't say the majority. The most common is for people to have four sort of trees. So this would be a tree with actually a forest with four connected components, which is why they're differently colored here. But there are people who have three, and there are people who have as many as six. So it varies. There you go. It's biological variation. So, and this has actually been a problem because one of the types of analyses that people would want to do is something that is trying to analyze the combinatorics of this tree. And I'll say a little bit more about this later. But the fact that you can have differing numbers, I say tree, even though it's not really a tree, but the fact that you can have different numbers of major branches makes comparing the combinatorics between patients kind of not really workable. And we have a precise sense in which it's not really workable. But that's a good question, actually. Are there any other questions? Okay. So let's do this analysis. So here's the data structure. How is this actually reconstructed? So let me go back to these images. So what they did to get this picture was they did a tube tracking algorithm, which basically, I'll just show you the data structure, and then you can figure out how they did it, I suppose. But this has the data structure is that we have a bunch of spheres. So those tubes that I showed you on the previous slides, those were actually just unions of balls. And what they do, each ball has a radius. And then what you see is literally just the union of all of those balls. Now, this is a much sparser picture than the actual data set, and I also haven't included the radii. So a priori, there should be a radius of this thing, a radius of, and also in the actual data, these points would be really, really close together. So between here and there, there'd probably be like 100 sphere centers. And so it's really densely packed with spheres, so it really looks like a tube. but in order to convey what the math is going to do in the math and statistics, I've drawn it a lot more sparsely so that you can see what's happening. Okay? So here's the data structure, and the idea is to do persistence centered on the branch points. So what we're going to do is we're going to take xt to be the set of points in x. So x is the blood vessels. So we're going to take the set of points, by which I mean sphere centers. We're going to take the set of sphere centers in our blood vessels that are within T of some branch point, or really the set of branch points. So I'm not picking only one branch point. I'm taking the set or a set of branch points and taking all the points that are within T of that and saying that's what xt looks like. So I'm going to show you some xt. So this is what? The Euclidean metric. This is just it's a bunch of blood vessels in 3 space, and I'm going to. Yeah. So let's look here and see what happens. So the question is in T in which metric, and so the question is if the artery comes back really close to a branch point, what do? Let's watch and see what happens. So this is T equals 0. We get or less than 0, so we get nothing. And then when T is very small, positive, then we get everything within epsilon of this branch point. Right, everything inside this red circle, and now the red circle is going to start enveloping more of the tree. So now we're going to get that one extra point above it, and now we're going to envelop it a little bit. Oh, I don't know why that's bouncing around so much. We're going to envelop a little bit. A little bit more, and it's going to get these two. A little bit more, and then it's going to get just this one. A little bit more, it gets that one. A little bit more, and now you'll notice it's got this one, but not the one in between. So anyway. There's going to be some better pictures in a second. This is just to give you an idea what I mean by xt. It's the stuff inside of the red circle as the red circle grows. So at this point, it has 3 components. Yes, it has 3 components. The 3 components are this one, everything in here, and then this little thing up there. Yes, yeah. And so we're going to see that really clearly in a second. And so on. So now what I'm going to do is I'm going to draw the blue thing, but it's going to be enveloped by this red circle, so I'm going to draw red for the stuff that's been enveloped. So first you start with nothing, then you get just the 1 point, then you get that neighboring point, then you get a few more neighbors, and as the circle grows, you get more stuff. Aha, and here's the point at which we were looking at the 3 components. I guess there were 2 vertices inside of this component and not just one, so I didn't read it right, but that's fine. And then you keep growing. And the point is that you should record the birth time of each new component. So time is the same as radius, I guess. So the birth radius of each new component, and then the death of each component. And a component dies when it joins an older component. So this is, as Adelsbrunner likes to say, this is killing the children. So the most recent one to be born is the one that you attach to the older one, and the older one is viewed as persisting for longer. So you'll notice that with the very slight bend that occurred up here, there was a new component that appeared and then immediately joined to the rest. Whereas the one down on the bottom, it's been created and it persists for a little while, persists, and then it's finally joined. Sergey, does this answer your question? Yeah. So when something bends in close to a branch point here, then it ends up persisting for a long time. So this is meant as a sort of a measurement of some kind of bendiness of the blood vessels. And there are medical reasons why we want to do this. Various diseases involve changes in vascularization. So cancer, for instance, involves very bendy blood vessels. And this is sort of a precursor study to see whether or not persistent homology might be something that we could use as an automated way to look for things like brain cancer from scans, non-invasive scans. As I say, this is easily computable, and I want to show. So this was a heuristic picture. What I want to show you now is the actual picture of what you get when you do this thing on Steve Marin's brain. And let's use this one, like that. So what's going to happen at the very? So there's a little tiny red dot, but ignore that. So now there are very small balls growing at the branch points. Now we're not using all the branch points. What we found is that if we use only. Is it stepping more? Weird. If we use only the branch points in one of the quadrants of the brain, then we end up getting data that have better correlation with things that we know that there kind of should be correlation with. So what this looks like, and it's going to be complicated looking, is that these balls grow, and cleverly Sean, who was the one who put this video together, made the balls get more and more transparent as they get bigger, and at a certain point they just vanish. And now you can see the red sort of enveloping everything. All right, so if I let this go, you'll see the red envelops, and then it's enveloped the whole thing. And now it can, I'll just rotate it backwards. There's a little bit of, since it's not drawn in perspective, it sometimes rotates the wrong way, and it feels pretty uncomfortable. But okay, let's see. So, so for instance, you can see here. I want to show you just an example of this of persistence. So if you look over here at this little edge here, something just showed up, and then these two things are going to persist for a little while, and then they'll join each other. And now they're one component that's persisting for a little while, and now it's going to keep going and join with that one, and then finally it will join with something bigger, and then so on and so forth. So that's what that, if you remember that barcode that I showed you with all the purple lines, that was, I don't think it was definitely this, actually it might not have been this brain. Anyway, it was one of the data points from the study. We have about 100 of these things. Okay, just for fun, let's just go through that once more. All right. And they shrink. Oops. Okay. Back to this. So when you, whoops, let's make it bigger again. So when you try to do statistical analysis on this thing, there's a number of ways to go, but what we did was some sort of principal component analysis. And there's a number of ways to reduce it to a principal component analysis. One way is to use multidimensional scaling. There are ways of taking sets of persistence diagrams. Actually, this is kind of interesting from the point of view of this conference, actually. So, this conference has something to do with, think about moduli spaces of representations, either, just the sets of quiver representations or moduli spaces, when you mod out by group actions and so on. What we did here, or what these authors essentially did, was they looked at the space of persistence diagram. So this is a shadow of quiver representations in some sense, and they made that space into a space, they made that set into a space where you can actually do statistics. They showed that there's a metric where you can, which has good enough properties that you can do statistics on this space. Anyway, so multidimensional scaling is a way of trying to fit all the data points, which are points in this space, into a Euclidean space. Once you have them fit into a Euclidean space, now you can do principal components analysis. Take the first principal component, and you end up being able to get some correlations with things. Why did we use principal components on only the 5 longest bars? Well, you might have noticed that very, very short bars are noise. Because if you have a lot of sample points, then you expect that when you grow them a little bit, a bunch of little components are going to join together very quickly. And you might think that, oh, well, maybe there's a sphere, maybe there's a bit of a bend over, some wiggling something over here, and you have a sphere that's expanding to envelop it. Well, it might envelop little tiny bits of it first, but very quickly when the sphere gets big enough, they'll all join together. So very short bars are really noise that you can ignore anyway. And so what we did was we analyzed only the 5 longest bars. We actually looked at a range of things and found that the 5 longest bars gave us the best signal. The conclusion that we drew is that longest, if you look at the 5 longest bars, then in older patients, the longest bars, they tend to be shorter than they were in younger patients, and they also tend to occur later, which means further up and to the right in the diagram. And there are good biological reasons for this. As you age, the blood vessels the thicker parts get thicker and the thinner parts get thinner, and so you expect that the part that's seen on the MRI it gets sort of stouter, and that would give you this kind of effect. And the correlation between the persistence and age is pretty good. So this says that if you try to look for linear correlation between what the persistence diagram looks like and what the age of the patient is, then the Pearson correlation's 0.46. 0.5 would be a very strong linear correlation. Maybe 0.2 would be a fairly weak but still present linear correlation. 0.46 is quite good, and we're quite sure that the correlation is there. So previous analyses have the best correlation they've gotten is this 0.25, which says that there is a correlation, but it's not clear. And as I say, sort of biologically speaking, there should be a correlation, but it's not quite clear how to get it from a statistical analysis. Anyway, discrete methods, these are methods that, that ignore everything except for the combinatorics. And those have not been so successful with these brain trees because sort of it's the geometry that's really important and not so much the exact combinatorics. It's sort of a statistical type of combinatorics, which this doesn't really do well. Dict paths, this is the standard dict path encoding of a planar tree. So the first thing you have to do is flatten the tree into the plane. You can remember the edge lengths, but you don't remember anything about the geometry. And that's why we think the correlation isn't quite as strong. But since the phenomenon that they're detecting is something about lengths of vessels, it is getting something. And now the phylogenetic tree analysis. So what we did at the SAMC working group was we one of the authors, one of the people connected with Steve Marin at UNC, had put landmarks on the cortical surface, and you can use those as the leaves of a phylogenetic tree by just sort of joining each landmark to the closest point on the tree and then pruning all the other leaves. And then you end up with a phylogenetic tree, and you can study, you can try to do statistics with phylogenetic trees, and what you find is that you get less than nothing. You find that, for instance, the average of these trees is the tree that has zero information in it. And you can think that, oh, that's meaningless, but actually it's meaningful. It just doesn't give you any, the information it gives you, it says something like the combinatorics is sufficiently spread around tree space that you're getting no information by viewing the by looking at the combinatorics of these brain trees. You're better off doing something. This is what really convinced us we need to do something that involved the geometry as well as the combinatorics. Yeah. Yeah, so that was what's the perceived medical explanation for the correlation between the way the persistence diagrams are distributed and the way the ages are distributed. It's that the blood vessels, as I say, the thicker parts get thicker and the thinner parts get thinner. So so when you age, there are partial blockages which cause the blood vessels to want to get wider so that they can still get the same amount of blood. But then the capillaries sort of infiltrate sort of more thinly, and so they're sort of more dispersed. And so you end up the thinner parts get thinner and the thicker parts get thicker. And I mean, I can maybe take a couple more minutes to explain. I could take a couple more minutes to explain really why you expect this to be what you get from that. But it's reasonable. That is, we can make up a story that's consistent with what's already known about the way blood vessels in the brain age. We don't know whether that's the reason, but at least we're not shocked that that we had a correlation that was this good. Yeah. Yeah, Sergey.
Wouldn't it be simpler to just take some fixed points inside the brain, like the middle of it, and start growing from them, not necessarily from your balls?
Yes. Yeah. So wouldn't it be simpler to take a fixed point in the brain and just start growing from that? And the answer is we're actually doing that. That's one of the things that we're doing next. I don't know if it's going to make it into this paper or the next one. But that is definitely something we're going to try. There is the problem of picking a consistent point in the sense that when you have two brains, how do you say what's the corresponding point in the middle of this brain to the corresponding point in the middle of this one? Now, the landmarks that were done by Ipek Ogush, those were painstakingly placed on each of these 109 brains. 128 landmarks on each of 109 brains, and every single one of them placed the landmark by using biological features. Now, there are other ways of automating so you can sort of, these are really cool videos. You essentially do flows on the space of diffeomorphism group of R3 that take one brain into the other brain, and then that allows you to get an average brain. And you can pick one. No, I'm serious. You can see that Sarang Joshi has these videos. They're totally awesome. And you can flow one brain into the other, and then you could pick one point in the average brain, which, by the way, it looks kind of like a fuzzy brain because you're averaging a bunch of structures that are spread out in 3 space. Pick one point in there, flow it to all the others, use that, and then grow from there. But that is a suggestion. We are going to do it, but we haven't done it quite yet.
I would explain expect the outcome to be strongly dependent on the choice of the
It shouldn't be particularly strongly. Well, it'll be very different if you choose the point to be in the middle as opposed to on the side or something like that. So the distribution of how long the longest bars are, for instance, will depend on sort of how far you are from the extreme points and this kind of. Anyway, there are things to consider. But yes, we are actually going to try that and see what happens. We're also trying things like choosing the growing points to be on sort of on the fatter blood vessels instead of the thinner ones, and so it's not just looking at all branch points. By the way, we tried all branch points, and we essentially got no signal because all the bars were too short. That's another thing that we tried. We want to. One of the things is that we don't have all that much information about these. We know that they were all healthy, for instance. Sex is a good one. Actually, sex works. Yes, sex is a good one. Sex works. So did we use other parameters besides age? So we used sex. And in particular one with the landmarks, the difference between male and female brains, statistically speaking, is a difference is that the blood vessels, and I forget which is which, in one of the two end up, the thicker parts are farther from the cortical surface in one than the other, statistically. And I don't remember which is which. Or which would be better. Yes, Greg.
So this is more biological, but would you expect that if you started the balls growing in different regions of the brain that you get different behavior with the longest bars?
If I start growing the balls in different regions, would I get different behavior for the longest bars? Somewhat, yes, but I don't know whether it would be detectable. I mean, in some sense, these are blood vessels and not neural connections. So this is more just how the brain is fed rather than how it acts. And so I'd kind of expect it to look, roughly speaking, the same. I wouldn't know what differences to look for, but I would expect some differences. I expect that some differences might be detectable, but I wouldn't know what to make of them. Yeah. Others. Yeah, so in fact, the question is if you're looking for cancer, then you're looking for tortuosity, the bendiness of these blood vessels. Yes, it is possible to define measures of tortuosity directly, and in fact, Liz Bullitt has done that with her group. And they actually do use these things, or have used them to say that we can actually tell statistically with reasonable accuracy that this is a this one is a cancer and this one is not, for instance. Now, or this one's. Say it again. Okay, so this what are the advantages? This one would be something that you could use to look for it when you didn't already know that there was something you were looking at. You wouldn't want to use the other approach when you're doing a screen. You would do it when you're trying to take something that you think might be, and then you want to run a test on it sort of specifically. But this one for the moment is sort of a really it's not at that stage yet. This is proof of concept. Can we get anything statistically interesting out of persistent homology when applied to these things? The answer so far is yes, and then it will take a lot of development to see if we can get some actual applications of that sort. Yeah. Okay. So on to fly wings. So these. I should mention the first. Now I guess it's now about 2/3 of the talk. Has been standard applications of applications of standard persistent homology. There was no new math there, although there was this way of presenting it to this audience. But anyway, there's no new math there, and the goal was really to then get to this part where I'm going to tell you about some new math that we want to do in service of biology, and new biology that comes in service of the math. You'll see that a little bit. So these are normal fruit fly wings, Drosophila melanogaster. These are the little buggers you see eating your plums in the summer. You should be looking at these and going, yeah, those look roughly the same. And they should look roughly the same to you. They're slightly different shapes, but they look roughly the same. And these are topologically abnormal wings. So I don't know if the screen is bright enough. Can anyone see any differences between? Yeah, Alan? Which one? Oh, it is bright enough. Okay, good. Can you see the topological differences between some of these things? You can pick any of them. Like, what's the difference between, where's the error in this wing? Yeah, exactly. It's a little triangle over here. There's an extra vein. No, someone was making the little triangle. Yeah, that's right. How about this one? This one's actually fairly easy to describe. Yeah, the bottom vein doesn't quite touch down there. And similarly, this one has an extra sort of doubled thing up here, and this one has another sort of doubled thing up here. And how about this one? This one's the easier one to see, actually. Yeah, it's got an extra bar over here. And this one has an extra bar between the two veins up there. So these are caused by genetically induced changes. That is, in the lab, they know they force the genes to be mutated so that the flies would grow with, well, they wanted to see what kinds of effects. And the idea is now to, the goal is statistical analysis, and you want to understand not just, so the previous analyses they've done have been like, okay, so if we look at genetic variation, then let's look at the changes in the shape. When the topology stays the same, you can look at changes in shape, and there's a notion of a shape space, and that's sort of a relatively standard type of statistics to do. But when there's topological variation, how are you supposed to include those in your statistical analysis? What you really want to be doing, so you want to give appropriate weight to these new singular points in addition to the varying shapes. And you want to compare the phenotypic distance to the genotypic distance. That is, I can tell you which genes you have to change to induce some of these changes, but I also want to then compare those sort of level of change to the level of change that you get on the phenotype level. And then what we're trying to do now with the geometry is measure differences in phenotype in some kind of consistent way. So what you really want is to place each of these wings as a data point, and you want to place them all in one space where you can say, okay, the distance from this one to this one is that, the distance between these two sample points is that, and then do statistics using those various distances. But it's not so easy to place these things in a single space. So what are the obstacles? Shape spaces need constant numbers of landmarks. So what a statistician means by shape space is actually what you mean by a vector space, or a space of matrices modulo a group action. So you take a bunch of vectors in a real vector space, and you say that two collections of vectors are equivalent if they're the same up to rotation, or the same up to rotation in translation, or rotation, translation, and scaling, and these kinds of things. And then the space of shapes, which is the quotient by that group, is the space that you're sampling from. And as long as you have constant numbers of landmarks, that is, as long as your matrices start off being all the same size, you can do geometry on that space, metric geometry on that space, and people do metric geometry on those spaces. But here we have different numbers of landmarks. The landmarks that we're using in the wings would be things like the interesting branch points, where the veins have interesting topological structure. So now, stability. So you could try using persistent homology by just say sampling, taking a bunch of points sampled from the fly wings, from the veins, and then growing the balls around them. The thing is that tends to give. So there's a notion of stability for persistent homology that says that if you take a whole bunch of take dense sampling from a space, and then if you wiggle the points a little bit, then the persistence diagram wiggles just a little bit. And so if you remember these wings that had the fairly small in scale but big in structure changes, persistent homology, when you sample from these veins as opposed to sampling from these veins, this feature is very small, metrically speaking. And so the persistence diagram for sampling from these veins and sampling from these veins will look very, very similar to each other, provably, in the sense that the persistence diagrams will look similar because of the stability of persistent homology. So in that sense, dense sampling from you can't just sort of sample from the veins and look at persistent homology there, because it will understate the new topological features. So it won't, the genetic variation will not be reflected in your measure of the phenotypic variation. So you need a better measure of phenotypic variation. So the first attempt that we thought of was, let's just use brain. Yes?
The map of phenotype is well understood?
No, it is not. There are a lot of things known about it, and I'll get a little bit more detail about that. But in general, these things are not well understood because, for one thing, genotype changes tend to be discrete, and phenotype changes tend to be continuous. And there's a lot more than just genotype that affects it. There's metagenomics. There's the embryonic environment and these kinds of things. So there's a lot of factors that come into it. But the goal is to understand, let's say, the extent to which one can predict or correlate the phenotypic change with the genotypic change. Yeah. So a lot is understood, and I'll get a little bit more into that. So brain tree persistence, you can go around the singular points, which is a great idea. So for instance, for each new source point, you get a new source point for each new singular point. That is, you add an extra ball whenever you have an extra crossing, and that's actually a reasonable thing to do. it does understate the new singular points that are too near the old ones. So the same examples I was pointing to before, when you have the little triangle cut off, your new point, your new growing balls are really close to the old ones, and so the persistence diagram is still going to look pretty much the same. It'll just have an extra little short bar in there somewhere for those balls to join each other, basically. So it's good that you have one new source point for each singular point, but bad that it understates the new singular points when they're too close to other ones. Another approach you could use persistent intersection homology. So Bendich and Hare invented persistent intersection homology, which is really just intersection homology, and noting that the conditions that define it are compatible with the way that you actually compute persistent homology. So there you only, instead of using all simplices, you only use the proper simplices. In our cases, a proper edge, and these proper conditions have to do with the perversity. They say that instead of requiring that the intersections be transverse, you require them to be transverse, except there's some allowance for non-transversality, and that allowance is called the perversity. In this particular case, since we're dealing with just graphs and things, that means you're only looking at proper edges, and edge is proper if neither endpoint lies in the zero stratum. That is, neither endpoint is a new special intersection point. The problem... So when you're dealing with such small dimensional things, there aren't that many choices of perversity. But the answer is you do whatever the biology wants you to do. That's the goal that we're after. Is we want that the metric that we choose be biologically the most meaningful. And these attempts, they're perfectly good attempts, and you can do analyses using these things. It's just that they don't quite put their finger on where the biology is telling you that the distinction should be. And in this particular case, with persistent homology, this is the space you're looking at, and the blue edges are the proper ones. These green ones are not proper because they use this singular stratum. And the problem is that now the new features, what they're going to do is they're going to forever disconnect the space. There's going to never be a way for you to get from this part of the vein to that part of the vein using proper edges. And so in this sense, the new features are going to be given infinite size. Now, if you could somehow go around those new features like this, then you might be able to have them have finite size. And that's what we're going to do. That's what we're going to do on the next slide. So that's persistent intersection homology, and now we're going to go to, I guess let's call it stratified persistence. So the idea that we have is that, well, when you look at an image, right, you're thinking that that a fly wing it's a two-dimensional thing with mostly a two-dimensional structure with some one-dimensional stuff running through it and some special points. Now that's in some sort of faraway sense true, in the sense that when you look at it from far away, that's what it looks like. But in reality, this is just a close-up of one of the images that I had on a previous slide. These veins are not one-dimensional. They're two-dimensional. The intersection points are not zero-dimensional. They're two-dimensional. So there's a true sense in which the entire image really is all two-dimensional. It's just that we're declaring some of these pixels to be of dimension 2, to lie in a dimension 2 part of the image, and some of these pixels to lie in the dimension 1 part of the image. This is dimension 2 part of the image. This is the dimension 1 part of the image. This would be a dimension 0 part of the image. And the idea here is now to actually, let's be realistic and grant each stratum an appropriate thickness. So instead of requiring that you lie on something that is zero-dimensional or one-dimensional, say, you can lie as long as you lie within some fixed amount. And so then assign that thickened stratum, which is now full-dimensional, but you're going to pretend, from the point of view of calculating persistent homology, that its dimension is the stratum dimension that it came with, even though it's actually, as a set, full-dimensional. So you're just going to pretend that the dimensions are what they started off being. So then there's this notion of delay function, which is the metric analog of perversity, and the idea is that for each stratum S, you have some start time A to S. So remember when we had the balls in the brains, what we did was we just put all the balls there and we started growing them. And we saw when various parts of the brain got enveloped and so on and so forth. So what we're going to do here is say that for each stratum, we might have a different start time. Now, there's a condition that you need, and it's this compatibility condition over here that prevents very counterintuitive things from happening. But you want that the lower dimensional strata start expanding before the higher dimensional strata do. Otherwise, you end up with very strange things. I'll leave it to you to figure out what kinds of strange things. But the idea is now that you just stratify R to the n by saying that you look at your point and you say, this point is going to be associated to the stratum corresponding to this vertex if that vertex is the first stratum to envelop that point. And so, for instance, and the idea is to put some sort of appropriate thickness on the stratum, where the appropriate thickness comes from sort of biologically motivated. How thick should each stratum be? So in this particular picture, it would be something like this. So this point starts growing, and then this point starts growing, and then these 2 points start growing, and then these 2 points start growing, and then a little bit later, the edges start to grow. And so you'll see what happens is then that this point that's in here will be enveloped by this vertex before it's enveloped by either of the two blue edges. So this purple stuff all gets put in the zero stratum. Yeah.
What do you mean by S plus equal as a T whenever S intersects T?
Intersects T bar, T closure. Yeah, so it's very important that we're saying that this zero-dimensional stratum does not intersect this one-dimensional stratum, but it does intersect the closure of this one-dimensional stratum, whereas this blue stratum does not intersect the closure of this point, which is just the point itself. So this... Yes. So I'm saying yes. So the question was, what does it mean when S intersects T, and the bar over it's important, and the conclusion is that pretty much you want to have the faces there before you have the higher dimensional strata that you're inserting. Yeah. Now, when you do this, the conjecture is that that the persistence diagram is stable with respect to the choice of eta. That is, as a biologist, as a scientist, you're never going to get exactly what is the correct eta to put there. What you want to know is that if you've got something close, that if you wiggle the choices of start times for each of the strata, that the persistence diagram that you get out from it won't change too much when you wiggle the start times a little bit. And this is something that we have yet to prove. We think that it's true, but for all we know, maybe its truth depends on putting a few more conditions on what it means for the delay function to be just more conditions on a delay function in order to get that stability. We don't actually know. But in order to get meaningful statistics, this stability has to be there. Otherwise, you're doing something really arbitrary, because you could have chosen a slightly different eta, and then you'd have different looking statistics. Yes, Sergey? Sorry, you can prove so the theorem, this conjecture is a conjecture. So persistence diagram is stable with respect to the choice of eta. What that means is that persistence diagram is a continuous object. It's a bunch of points. So one way to draw a persistence diagram is just to plot birth time, death time. Or you can view it as a multi-set of intervals, if you like. I don't care how you want to view it, but it's an object that's basically a planar object. When I choose a slightly different eta, I'm going to get a slightly different persistence diagram. And I want that the persistence diagram as a function of eta is continuous, as long as you don't do things, as long as you don't make topological changes, such as, for instance, clearly if I make these two purple balls grow so much that they start overlapping, I'm going to get a different looking persistence diagram. But I'm not allowing you to do that. I'm saying if I grow this one a little bit and shrink that one a little bit, the persistence diagram won't change too much. It's a continuity statement. It says that the persistence diagram is continuous as a function of the choice of eta. Yeah. Okay, so future biology. So this was the biologists asking the mathematicians, gee, I'd really like to do this. What kind of math do you have for me? And the answer was, well, we have some math, but the off-the-shelf stuff doesn't work, so let's invent something. And now we have some conjectures to prove. But the response to the biologist, you can step back and say, well, the math would be better if you could give me data that look like this instead of what you're actually giving me. So what the biologists are currently giving us is they're giving us the end result of a genetic change. They're giving us a genetic change. They're growing the flies up. They're looking at the adult wings, just sucking them into a vacuum and taking a picture and so on and so forth. And that's giving us topological objects that are quite different from one another. But that's not how genetics gets turned in. That's not how genotype gets turned into phenotype. There's a process that's involved. And if you could follow the process and say that the process of getting from this genotype to that phenotype compares to the process of getting from this genotype to that other, this other genotype to that other phenotype, and compare the processes instead, you might get a more meaningful answer. So I asked this question to David Houle the first time I met him, basically. I said, can we do this? It's important. So vein formation, really almost all these types of developmental processes have to do with there's some gene that's expressing a protein. You look at the level of that protein, and whether or not a cell decides to turn into a certain other kind of cell will depend on some sort of thresholding. Is the expression level high enough, low enough, whatever it happens, in some range, that kind of thing. So what one can do, and they can actually do this, and that's what these images are. These images are not so easy to see, but they are. You get the fly embryos to glow proportional to how much of a gene they're expressing. So you can measure the amount of gene expression cell by cell in a fly wing embryo. In fact, they know, it's kind of scary, what happens to basically all of the 50,000 individual cells that become the fly wing. They can follow them throughout development in some sense. They know what happens to them. Now, they haven't tried to do that in a systematic way, but that's the thing that I asked. I said, can you tell me what the expression levels are in each of the cells throughout the development of the fly wing? And the answer was essentially, yeah, we can pretty much do that. It would be a lot of work, and we haven't done it yet, but it is possible. So I said, okay, let's do that, and let's, instead of comparing the end results, let's compare the actual time series. And now you're comparing things that start off in the same place, and then they diverge, and you can measure how far apart those sort of sequences of functions are, as opposed to measuring how far apart the end topologically different veins are. This is sort of like the same way that theoretical physicists handle the problem of singularity in space-time histories of point particles. You have a point particle, it's got a space-time history that, it's one particle and then it diverges and now it's two particles, and you have the singularity at the branch point. And so their solution is to say, well, let's up the dimension of the particle by one, and now you have a string, and when it diverges, now you have a nice pair of pants. It's not a singular thing anymore because the place where it was singular has been upped in dimension by one. And we're doing the same thing here, except so physics already has one space and one time dimension. We have two space dimensions and no time dimension. By throwing in the time dimension, we're sort of removing the singularity. And instead of looking at the sort of comparing two end discrete things, we're now looking at the time series, and now that should be a continuous change from one. When you sort of tweak the genetics so that the expression levels change a little bit, you're going to change the time series a little bit down here, and that change in the time series will grow as time goes on, so you can see where the divergence occurs. Anyway, so this is a suggestion for what the biologists should do, and I'm seriously contemplating whether or not I should sort of enlist people to do it for real, possibly in David Houle's lab. So this is something that would make the mathematical analysis better, but would also make the biological analysis better, because you are more directly connecting the phenotype to the genotype. You're actually looking at the expression levels and not just at the end result phenotype, but we're treating the phenotype as not just the adult phenotype, but the entire process of development. And so it gives you a sort of biologically more meaningful answer. And another thing we could try to do with this is to reconstruct phylogeny from morphological measurements. People do that already a lot from genetic measurements, but it would be really interesting to try to do that sort of thing from phenotypic measurements. Okay, so I'm almost out of time, so let me just go through just some general, what have we seen here? So the rationale is that when the space of data objects or each data object itself is stratified, in the case of, for instance, a tree or a graph, or something like that, it pays to take special care of the geometry topology, and I haven't talked about this one. It's sort of an entirely other talk. The asymptotics at or near the strata of low dimension. Just to give you an idea of what I mean by that, asymptotics. So brain vasculature, each data object is a stratified space. It's a tree. When you're talking about fly wings, the idea is to construct a metric space which is stratified. And you want that to be a stratified metric space of data objects. And further examples, these are sort of when you're sampling, your space of data objects is a stratified space, and you're trying to sample from it. You can still try to do statistics there. Like the way that you do a central limit theorem, it gives you limited distribution on Rn. You can do the same thing when you sample from a stratified space, but it gives you some non-intuitive answers. And the spaces that have been looked at so far, mostly they're all related to tree spaces. So open books are just bunches of pages glued along a subspace. When you're sampling from a binary tree or a space that is flat so a two-dimensional space that's flat everywhere except for one point where the curvature is negative, so the angle sum is bigger than 2 pi, and the tree space T4. Anyway, when you sample from these things and look at the asymptotics, you get answers that are funny, and they depend on the geometry of the space near the singularities. And there's some hope that so these sticky questions about sticky central limit theorems and such, there's some hope that you can try to understand, you can get invariance of singularities in stratified spaces by looking at the asymptotics of sampling nearby those singularities. That's a very general type of statement which I don't expect anyone to make too much of because I haven't talked about it much. But that's one of the questions that we've been working on, motivated from the biology. When you sample from a stratified space, what can you learn about a stratified space from sampling nearby the singularities? Essentially, that's what these 3 questions are meant to ask. And these persistent problems, these are ones that I've already mentioned, but we want to use persistence to detect pathology. That was the question that came over here. How does this compare to the other methods for detecting cancer, for instance? And also it gives you this conjecture. You want to get this stability for stratified persistence. And then this developmental dream, we want to. This is where to go with the biology. I would really love to see someone take these time series on the developing fly wings and then run some sort of analysis. It doesn't have to be persistent homology, although that's a good choice for it, but some sort of analysis to compare these sort of higher dimensionalized versions of fly wing veins to see what the connection is between the genotype and the phenotype. Okay, so I have some references. You can look through these, but with that, I'm going to stop. Thank you.
Any questions?
Yeah.
So, delay function, you actually get along with diversity. What it looks like is that it's giving matters tubular neighborhood data for working stratification. Should you really think of it as diversity?
So the question is, it looks like that these delay functions are giving tubular neighborhood data from Mather's work, and should I be thinking of it as perversity? There's 2 things. The first thing is, does it give me what's the tubular neighborhood information? Yes, it is. But the question, do I want to think of it as perversity? And the answer is yes, I do, even if it isn't. The point is that what it's doing is it's giving me rules about which simplices are or are not allowed to interact with each other. And it's doing it instead of in an absolute sense that this simplex can never interact with that one. It's giving me some sort of metric modification of that says, well, at small times, this one's not allowed to interact with that one. But when you progress time enough, then this one can interact with that one by going around the singularity in a way that perversity wouldn't allow me. But perversity, it wants to bar it's sort of a mix between the two. But yes, I want to think of it that way because it's basically because perversity gives me rules that are compatible with persistent homology. And we've had actually a bear of a time thinking of ways to soften the rules for perversity to give us something that we can use that bars the intersections for some amount of time, but not forever. And so we do want to think of it as perversity, even if it's not, and that'll direct how we try to develop the idea.